Hello everyone, I'm Sovon Acharya, a PhD student from MIT Institute of Biotechnology, MIT University, Gurugram, India. I am pleased to present our work at SANBIX, a platform that promotes collaboration and knowledge exchange among the researchers in bioinformatics and life science across the global south. Today, I will be presenting our study entitled Isolation and Genomic Characterization of a Novel Proteus mirabilis strain indica. Antimicrobial resistance or AMR is one of the most pressing challenges facing global healthcare today. Recent estimates indicate that approximately 1.14 million deaths are directly attributable to AMR annually, while nearly 4.7 million deaths are associated with drug resistant infections worldwide. If current trends continue, AMR is projected to contribute to millions of additional deaths over the coming decades, posing a significant threat to modern medicine. Among the bacterial pathogens contributing to this crisis is Proteus mirabilis, an opportunistic pathogen commonly associated with urinary tract infection, catheter associated infections, and wound infections. Its pathogenicity is mediated by multiple virulence factors, including swarming mortality, fimbrial adhesion, urease production, and biofilm formation. Despite its clinical significance, genomic information from the Indian isolates remain limited, restricting our understanding of their evolution, diversity, and pathogenic potential. So, to address this knowledge gap, we isolated a novel clinical strain of Proteus mirabilis designated as indica and performed whole genome sequencing followed by comprehensive or comparative genomic analysis. Our workflow include genome assembly, annotation, antimicrobial resistance profiling, virulence factor identification, sequence typing, phylogenetic reconstruction, and protein-protein interaction network analysis. So our results reveals that strain indica poses a previously unreported sequence type, highlighting the presence of underexplored genomic diversity within Indian P. mirabilis population. Comparative genomic analysis identified multiple antimicrobial resistance genes, virulence determinants, and insertion sequences elements that may contribute to bacterial adaptation and persistence in clinical environments. Furthermore, phylogenetic analysis demonstrated that this strain indica occupies a distinct evolutionary position compared with publicly available genomes, suggesting the emergence of a unique lineage within the species. These findings underscore the importance of regional genomic surveillance for understanding the evolution and dissemination of clinically relevant pathogens. One of the most significant findings of our study was the characterization of the virulence associated interaction network using string and cytohopa. We constructed a protein-protein interaction network and identified RPM-A, RPM-B, MRP-A, and MRP-B as the major hub genes within the network. In particular, MRP-A and MRP-B encode components of mannose-resistant proteases like fimri, which play essential role in bacterial adhesion, colonization, and persistence within the urinary tract. The network also highlighted other important virulence associated genes, including URC involved in urease production, fimbrial genes such as MRPD, MRPG, MRPH, and MRPJ, and adhesion related genes including IREA, ATFA, and YTER. Importantly, overlap analysis revealed interactions among virulence factors, antimicrobial resistance genes, hub genes, and insertion sequence elements. This observation suggests that pathogenicity, resistance, and genome plasticity are governed by a highly interconnected genomic network rather than by individual genes acting independently. Such interactions may facilitate bacterial adaptation, persistence, and survival under antimicrobial pressure. In conclusion, we report the isolation and genomic characterization of a novel Indian Proteus mirabilis strain indica. The discovery of a new sequence type, together with the identification of key virulence subgenes, antimicrobial resistant determinants, and mobile genetic elements provide valuable insight into the evolution, adaptation, and pathogenic potential of this organism. Our findings highlight the importance of integrating genomics and bioinformatics approaches to understand emerging bacterial pathogens and reinforce the need for localized genomic surveillance to combat the growing global challenge of antimicrobial resistance. Thank you for your attention.